Dear students, in previous class we have seen the structure of chloroplast, ribosomes. Today we shall see the cytoskeleton, cilia and flagella, centrosome and centrioles. Cytoskeleton. If we examine the cell, within the cell there is an elaborate network of filamentous proteinaceous structures present in the cytoplasm of the cell, collectively together called cytoskeleton. See, what is cytoskeleton? Cytoskeleton is nothing but network of filamentous. Filamentous means filament-like proteinaceous structures. The network of filament is made up of chemical proteinaceous structure which is present in the cytoplasm of the cell. It is together we call or collectively called cytoplasm. Once again, network of filamentous proteinaceous structures in the cytoplasm collectively called cytoskeleton. The cytoskeleton take part in many functions of the cell. So, many function means what? It gives mechanical support within the cell. Mechanical support. It helps in motility of the cell. Motility means the movement of the cell. Then it also helps in maintenance of shape of the cell. So these are some function of cytoskeleton, that is mechanical support, motility and maintenance of shape of the cell. Okay, that's all cytoskeleton. Now we shall see the cilia and flagella. You refer the diagram. Okay, there is a diagram. Both, that is cilia and flagella, are hair-like outgrowths of cytoplasm. Hair-like outgrowths of the cell membrane. The cilia and flagella are hair-like outgrowths of the cell membrane. Cilium is singular, flagellum is singular. What is the difference between cilia and flagella? The cilia are small, which helps in movement of the cell. Flagella are long when compared to the cilia. The flagella also responsible for the movement of the cell. Okay. The prokaryotic flagella or bacteria also possess flagella. Prokaryotic bacteria possess flagella, but these are structurally different from eukaryotic flagella. Note the point. The prokaryotic bacterial flagella are structurally different from the eukaryotic flagella. Okay. The flagella helps in movement of the cell. According to the studies made by electron microscope, flagella are cilium covered with plasma membrane. The studies made with the help of electron microscope, flagella or cilium covered with plasma membrane. And thin central core called Auxinome. You can see the diagram. Auxinome possess 9 pairs of doublets of peripheral microtubules. You can see in the diagram 9 pairs. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 pairs means 2 each. Possess 9 pairs of doublets. Doublet means 2. Peripheral microtubules and a pair of central microtubules. See the diagram. So peripheral means marginal. How many? 9 pairs are there. Pairs of doublet microtubules. At the center, we come across only a pair. ಒಂದೇ pair ಇದೆ ಸೆಂಟ್ರನಲ್ಲಿ. Central microtubules. What is the arrangement? The arrangement of microtubules we call 9 plus 2. 9 plus 2. The central tubules, see the diagram, are connected by bridges. Not only the bridge and also enclosed by the central sheath. You can see the red color. Central sheath is there. The central sheath which are connected to the one of the tubules of each. Now, the sheath is connected to the one of the microtubules of the peripheral one. You can see the diagram. Peripheral doublets by a radial spoke. You can see the radial spoke. Thus, there are also interconnected by linkers. You can see the interconnection also. Both Cilia and Flagella emerge from centriole like structure called the basal bodies. Very important for the NEET or CET examination. That is, cilia and the flagella emerge from the centriole like structure called the basal bodies. See the diagram now carefully. You examine section of cilia or flagella. You can see plasma membrane, the outer membrane. You can see. You can also refer peripheral microtubules. Microtubules, they are in doublet. ಎರಡೆರಡಿದೆ ನೋಡಿ. ಅಂಥದ್ದು ಎಷ್ಟಿದೆ ಅಂತ ಕೌಂಟ್ ಮಾಡಿ. 9 ಇದೆ. 9 doublets of peripheral microtubules are there. ಕ್ಲಿಯರ್ ಆಗಿ ಕಾಣ್ತಾ ಇದೆ. That is the blue one. In addition to that, at the center ನೋಡಿ ಮಧ್ಯದಲ್ಲಿ there are only 2 central microtubules are there. ಎರಡಿದೆ ಮಧ್ಯದಲ್ಲಿ. ಜೊತೆಗೆ ಅದಕ್ಕೆ ಒಂದು ಶೀಟ್ ಇದೆ. You can see the red colored one sheath. ಆಮೇಲೆ ಮಧ್ಯ both are connected by a bridge. Blue color bridge is also there. You can see the bridge also. Then one of the microtubule is connected to the sheath, central sheath. That's also visible. The structure of cilia or flagellum under the electron microscope. Okay. You must practice this diagram. ಇಲ್ಲಾಂದರೆ ಬರೋದಿಲ್ಲ. ಪ್ರಾಕ್ಟೀಸ್ ಮಾಡಬೇಕು ಇದನ್ನು. Next one is centrosome and centrioles. Centrosome is one of the cell organelle present in animal cell, usually absent in plant cell. One of the difference between plant and animal cell. Okay. Centrosome contain two cylindrical structures. You can see now, cylindrical structures called centrioles. Now you can see the centrioles. There are two centrioles. These centrioles are arranged right angles to each other. See carefully. The centrioles are surrounded by amorphous pericentriolar material. Okay. If you examine these, it looks like a cart wheel. Cart wheel ಅಂದರೆ ಗಾಡಿ ಚಕ್ರ ಥರ ಕಾಣ್ತಾ ಇರ್ತದೆ. ಎತ್ತಿನ ಗಾಡಿ ನೀವು ನೋಡಿರ್ತೀರ, ಅದ್ರದ್ದು ಚಕ್ರದ ಥರ ಕಾಣ್ತಾ ಇರ್ತದೆ. ಸೊ ಮಧ್ಯ what we call it as central part is there. ಕನ್ನಡದಲ್ಲಿ ಅದನ್ನು ಗುಂಬ ಅಂತ ಕರೀತಾರೆ. ಅಲ್ಲಿದ್ದ ಎಲ್ಲ ರೇಡಿಯಲ್ ಆಗಿ ಹೋಗ್ತಾ ಇರ್ತದೆ ಸ್ಪೋಕ್ಸ್. It consists of 9 evenly spaced peripheral fibrils are made up of tubulin protein. Very, very important for neat examination. Each peripheral fibril is a triplet. That is also very important. The adjacent triplets are also linked. Peripheral fibrils are in triplet. The adjacent triplets are also linked or connected. The centrosome contain RNA, glycoproteins, and even the DNA. This is most important point for NEET or CET examination. Okay? The central part shows hub, which is connected with tubules of the peripheral triplets by radial spokes. Radial spokes means protein. Okay. Now, the function, very important part. Centrioles form the basal bodies of cilia and flagella. When we studied the cilia and flagella, we heard about the basal bodies. So, these basal bodies ಯಾವ ಥರ ಆಯಿತು ಅಂದರೆ centrioles actually form the basal bodies of both cilia and flagella. Next, another important function, spindle fibers form spindle apparatus. Spindle apparatus is formed during cell division in animals. That is. Then it also gives rise to axial filament of sperms. So sperm is having axial filament. Okay.